All right, so we started last week discussing some genetic terminology. We established the difference between heredity and genetics. We said the heredity is the transmission of genetic information from parent to offspring, and genetics as a branch of science that actually studies heredity. We introduced some genetic terminology. We distinguish between a gene and an allele. We know that genes are the inherited factors that we get from our parents, and then these alleles are sort of different versions of a gene, right, that can code for various phenotypic expressions. We talked about the locus as the specific place on the chromosome where we can find a gene, the locus. We talked about the genotype as the set of alleles that an individual has, right? Your genotype is your entire genetic makeup. We talked about the phenotype as the outward appearance of a trait, right? Of a characteristic. So this phenotype can be physical, like what I look at you and see. It can be physiological, which is how you function as a person, and then it can also be behavioral, right? So all of these factors in our phenotype are determined from our genotype. All right, so jot that down and have a point of recall for that. All right, I made the point that when we talk about inheritance and what's actually being inherited, okay, what you're getting from mom and dad is not a physical feature. You're not inheriting a phenotype. You are inheriting a genotype, a set of alleles. Okay, so organisms, individuals, do not transmit their phenotype to the next generation. You transmit your genotype, and then it is the genotype that ultimately influences the phenotype. Okay, so that's kind of where we left off at. We talked a little bit about dominant and recessive alleles. We said that dominant alleles are those that will be observed. That's what we can actually see physically, okay? Whereas the recessive alleles are repressed, right? Masked, right? So if we had a heterozygous pair of alleles, only the dominant form would be physically present in the phenotype, right? Are we clear about that? We have any questions about that information before we move on?
No, ma'am.
Awesome. So let's keep going today. We talked about this concept already of dominant and recessive. Let's talk about how we get these distribution of genes, all right? Principles of segregation, jot this down, right? So we said that each organism possesses 2 alleles for any particular trait, right? You're going to inherit a pair of alleles, 2. All right, you can have a heterozygous pair or you can have a homozygous pair. All right, so the 2 alleles that encode a characteristic separates or segregates when these gametes are formed, right? So one allele goes into each gamete. So we looked at this process of meiosis about 2 weeks ago, a week or 2 ago, and we looked at how there's sort of this random distribution or segregation of these sister chromatids, right? This process of segregation is happening as meiosis occurs, right? This is a completely random process. The probability of which allele you're going to get into which gamete is completely random, okay? These gametes are formed, right? Gametes are our sex cells, the sperm and egg. These gametes are formed. There's going to be a distribution of genes, all right? Concept of dominance goes back to this idea that we have a dominant and recessive allele. When 2 different alleles are present in a genotype, only the trait encoded by the dominant allele will be observed in the phenotype. This is known as the concept of dominance. Write that down. If I ask you about the concept of dominance on your exam, you should be able to intelligently discuss this concept. This concept tells us that when we have 2 different alleles, which we'll refer to as a heterozygous pair of alleles, right? For every trait, there are 2 alleles. You can have a homozygous pair or you can have a heterozygous pair. Whenever there are 2 different alleles present in your genotype, only the trait that is encoded by the dominant allele will be observed in the phenotype. That is the concept of dominance. Write that down for me. Okay. The concept of dominance. All right, so Gregor Mendel did a number of different crosses, right? And after you continue to do things over and over and over, you repeat the experiment, jot down the data, you start to notice patterns, right? So after years of investigation and a lot of recollection of data, we have what we know as these predictable patterns of inheritance. One of the breeds or one of the experiments that Gregor Mendel did was what we call crossing, right? These are intentional breeding experiments. A monohybrid cross is a cross between parents that differ in only one characteristic, right? So he's breeding these plants. They're differing in only one characteristic, okay, genetically, all right? So that's what we'll call a monohybrid cross, meaning one. Mono means one, all right? We're crossing two plants that are genetically the same and they differ by only one characteristic, and we're observing the outcome, okay? So the first generation of a cross, right, is what we'll call the parental generation. Those are the parents, right? And then the subsequent generations that are produced are offsprings, right? We'll refer to them as F1, first offspring generation, F2, filial generations, and so forth, right? So P stands for parental generations. So the first experiment that Mendel conducted was a monohybrid cross for the shape of the seed. That is the characteristic, right? So he had a homozygous pea plant for round seeds, right? And then he had a homozygous plant for wrinkled seeds, okay? He crossed those up and there was a likelihood of the outcome, okay? So the offsprings from the parents in the P generation is F1. That's your first filial generation, okay? That's the first set of offsprings, right? F1, just make a mental note of this when we're looking at our crosses. And again, this is not a genetics course. We're not going to go into all of the fine details. You'll have more time for that when you take genetics, but you should just kind of understand these concepts from a general sense, right? So we have P as the parents. These are the ones that will be crossed, and then offsprings will be created. So the first generation of offsprings from a parent generation is F1. That's the first set of offsprings. So Mendel set up these crosses to examine the shape of the seed. And he examined the F1 generation, which was the first generation produced of offsprings. And he found that in the F1 generation, right, from a monohybrid cross that's differing in only one trait, he found that they expressed only one of the phenotypes. All of the F1 seeds were round. So let's look at what happened here. In this monohybrid cross, and I do have an illustration for those of us that are visual, just stay with me. During a monohybrid cross, Mendel planted the F1 seeds. He cultivated the plants, allowed them to sort of self-fertilize and produce another generation of seeds. All right. In this case, both traits from the P generation, right, appeared in the F2 generation. All right. So all of the offsprings in F1 were the same. We have another round of fertilization, and now we're starting to see both traits from the P generation. Both traits emerged in a ratio of 3-to-1. So after years of investigation, continue to repeat this, continue to collect our data, after a monohybrid cross, where we had two homozygous parents in that P generation. So in this case, they're only differing in the color of these petals, right? In this case, we have purple and then we have white, right? All of the offsprings in this F1 generation came off purple, because what it turns out is that this one of the allele pairs in that P generation contained a dominant allele. Okay, so every individual in that F1 generation were going to have a dominant allele in that pair. Okay. However, in that F2 generation, now we've got the presence of a heterozygous pair of alleles where we have the dominant trait present and the recessive trait present. And all of the individuals in F2 in that second generation are going to appear in a ratio of 3:1. 3 purple, 1 white. Jot that down. Monohybrid cross. Okay. Monohybrid cross shows us that when we cross, when we're breeding, and these individuals are genetically similar in all but one trait, a predictable pattern of inheritance from this monohybrid cross would result in the following. This is what I want you to know. The F1 generation, they're all going to be the same. Okay, we've got a dominant pair of alleles and we've got a recessive pair of alleles. All of the F1 generations are going to manifest that dominant trait. In this case, it's purple for our example for these plants. F2 generation, these offsprings are going to present in a ratio of 3:1. 3 representing the dominant trait, and then 1% or, one piece of it, 25% of it will exhibit the recessive trait. Okay, the recessive. All right, so that's the result of a monohybrid cross. F1 generation, they're all going to have the trait of the dominant form of the parent. All right, F2, these traits are going to appear in a 3:1 ratio. All right, so that's a monohybrid cross. All right, principles of inheritance. Jot these 2 principles down. The first being the principle of segregation. Right, the principle of segregation says that 2 alleles separate when gametes are formed. We know that is true. Okay, again, we saw these in meiosis. Okay, complete separation, random distribution of these allele pairs. Okay, so when these gametes are being formed, 2 alleles will separate. That's the principle of segregation. Completely random, absolutely no control over which allele from which pair goes into which gamete. Okay, the concept of dominance. The concept of dominance tells us that when 2 alleles are present in a genotype, only the trait encoded by the dominant allele is observed in the phenotype. You already wrote that down. Okay, we're adding to that now with the principle of segregation. This is something that happens. It must happen. You have no control over it happening. Okay, these are 2 very critical principles of inheritance that we know to be true. The principle of segregation says that 2 alleles separate when these gametes are being formed, right? The concept of dominance says that we're going to observe only the trait that's encoded by the dominant allele in the phenotype, okay, whenever we have 2 different alleles present. That recessive allele that's present in that pair will be masked or repressed. Questions? Okay. So sex chromosomes. So we looked at meiosis already, right? So for the most part, our genes, right? We looked at that human karyotype and we saw the layout of genes and we saw chromosome pairs 1 through 22. And then we saw that set of sex chromosomes, X and Y, right? So for the most part, our genes are the most important sex determinant in eukaryotic organisms, right? So our sex is generally determined by sex chromosomes. Okay, but in some cases, environmental factors can exert control over an individual's sex, right? But typically, members of one sex have a pair of similar sex chromosomes. So for humans, that's going to be the X and Y chromosomes. Jot this down if you don't know it. Female sex contains 2 X chromosomes, XX. All right. So if you're looking at a human karyotype and you're trying to determine some information, various things that are present, one of the things that you should be able to tell is if whether or not this individual is male or female based on the combination of sex chromosomes that are present on that karyotype. Okay. XX is female. Male sex have 1 X chromosome and one Y chromosome. Okay. So for most species, the Y chromosome determines the maleness. Okay, so XX is female, XY is male. Write that down. Okay, except in very extreme cases, these are going to tell us the sex of an individual and what they would physically present as. All right, the Y chromosome determines the male sex. Okay. Sex chromosomes, X and Y. So what information is on these chromosomes? So the X chromosome contains genes that are required for both sexes to be vital and living and healthy. Okay. So in essence, the X chromosome is required for life. Write that down. An organism, an individual, okay, during conception and gestation would not fully develop. That will not be a vital, it will not fully develop in the absence of this X chromosome. Okay, so a fetus with the Y chromosome and no X chromosome does not survive. That's not a viable abnormality. So the X chromosome contains information that is absolutely essential for life. Thus, we see the X chromosome in both sex pairs. All right. A person with one X, but no Y will have the overall appearance of a female, but with defects, right? Remember, we talked about those abnormalities that may be possible when things deviate from the norm. So we're saying that a normal female, a person will present as a normal female, would have a genetic assignment of XX, right? If something happens during reproduction, this individual only inherits 1 copy of the X chromosome. She's physically going to present as a female, but with some type of defects, okay? The X chromosome is essential for life. A fetus that inherits only a Y chromosome will not survive. All right, only an X chromosome, only a Y chromosome will not survive. The Y chromosome, okay? The Y chromosome contains the DNA for 95% of male specific traits. Okay, so this Y chromosome is necessary for us to get the traits that are male specific. So we're talking about reproductive organs, penis, scrotum, those types of things that are specific to the male. They're going to be coded for in this Y chromosome. Okay, that is what's going to distinguish the male from the female. The genes that are encoded on the Y chromosome contain 95% of the traits that are male specific. Write that down. Okay. Everybody good here?
No.
All right. Couple more principles of inheritance. So when we inherit these traits, right, and we talk about these principles of dominance, recessive, nothing is completely foolproof. Okay. Nothing happens without exception. All right. There's this idea of incomplete dominance. Jot this down. Right. So the concept of dominance tells us that whenever we've got two different alleles present, the organism will manifest the trait that's encoded by the dominant allele. Right. Well, in some cases, dominant is not always complete. It's not always 100%. All right. There's a concept of incomplete dominance that can be observed in phenotypes. Okay. So incomplete dominance is basically when the phenotype, what we see is an intermediate between that of two parents. So for example, if we breed a parent, well, we have these plants here where parent one is red, parent two is white, right? This offspring is going to present as an intermediate of both. All right. You mix red and white together and you get what? Pink. All of the offsprings in this F1 generation from this particular cross right here are going to be genetically the same in F1. They're all going to present pink. So what we have is a phenomenon known as incomplete dominance. Okay, so neither one of these alleles, if you will, for red or for white presented in a completely dominant fashion. So all of the organisms in this F1 generation are going to appear pink. Make that make sense. Incomplete dominance. Okay. What questions do we have about incomplete dominance? Okay. Neither one of those alleles in those pairs in that P generation presents as completely dominant. Okay. Then we have what we call codominance. Write this down. Codominance is when there's a simultaneous expression of the phenotype of both parents. All right. So we've got these white camellia, we've got these red camellias, they're going to breed, and then this offspring is going to be a mixture of not so much of a mixture where we're getting red and white to make pink, but we're getting a simultaneous expression of both. Right. So you're getting a little bit of red and a little bit of white in the first F1 offspring of this breeding. This is codominance. You need to be able to distinguish between incomplete dominance and codominance. Okay. Codominance, there's a simultaneously expression of the phenotype of both parents. So you're going to see both phenotypes present in that F1 offspring, not a mixture, but the simultaneous expression of both. Okay, that's different than what we saw with incomplete dominance. You must be able to distinguish between these two. These are some very common and sort of reliable patterns, predictable patterns of inheritance that we're looking at here. All right. So incomplete dominance, when the phenotype in that F1 generation is a mixture or an intermediate of the two parents, whereas codominance says that the offsprings are going to simultaneously express the phenotype of both parents. Okay, so if this was a white and black situation here, then this would be a black and white offspring. All right. All right, pleiotropy. So we're still talking about the fact that genes control, what we see in the phenotype, our genotype. So pleiotropy says that there's an ability of a single gene to have multiple effects on the phenotype. Okay. And so we see this a lot in genetic disorders, right? So one gene can manifest itself with a number of different effects on the phenotype. Okay. So for example, cystic fibrosis. All right. One pair of alleles can cause a number of different symptoms. All right. People with cystic fibrosis can have abnormally thick mucus in many different parts of their body. So we've got respiratory issues, we've got digestive issues, we've got reproductive issues. All right. And this is all due to abnormalities in this one gene. Okay. So pleiotrophy says that changes or that one gene, a single gene, can have multiple effects on the phenotype. Jot that down. Okay, and so again, this is again sort of going back to what I was saying, that no effect or no gene has a complete, like nothing is absolute here. Okay. Okay. So pleiotrophy is that, again, this one gene can have multiple effects in the offspring. Okay. And then lastly, there's this phenomenon known as the norm of reaction. Jot this down. I want you to really understand this concept of the norm of reactions. And so, like when I was talking about the determination of sex, for the most part, our sex chromosomes are going to determine the sex of an individual. And for the most part, we have mapped the human genome, and we know like which genes are responsible for, producing this particular protein product, and like what the functions of each gene is in our body, right? We know that for the most part. Well, what we have to take into consideration is that we don't sort of live in a bubble, right? Like, yes, our DNA determines our phenotype and how we function and what we look like and all of that, but we don't exist independent of our environment. So we have to take into account the role of the environment on an organism's existence, right? So the norm of reactions tell us that genes interact with their environment to ultimately influence what the phenotype of that organism looks like. So an excellent example of this norm of reaction, right, are seen with hydrangeas. The norm of reaction tells us that the range of phenotypic possibilities can develop from a single, so the norm of reaction tells us that there is a range of phenotypic possibilities. So there is a range. There are variations that can exist in the phenotype based on a single gene when you take into account the influence of the environment. So the hydrangea plants, okay? These plants are genetically identical. They have the exact same genotype. But if you plant these seeds in different soils, different exposure to light, in this case, different pH of the soil, these plants will all present with a different color. So the hydrangea, due to the norm of reaction, right? When we take into account the environmental factors that can influence an organism's phenotype, the color of genetically identical flowers can range from blue to purple to pink, depending on the contents of the soil before the flowers are planted, right? So if we again walk back everything that we already knew before we got to this conversation about the norm of reaction, we have an idea of what this genotype should look like phenotypically, right? What color should this plant be based on these patterns of inheritance that we know to be true? This gene, this allele will code for this trait. However, genes do interact with their environment to shape the phenotype, right? We see this very characteristically in the hydrangea plant. Okay, you've seen very beautiful hydrangeas. Some are purple, some are pink, some are blue. Okay, but we're saying that these genetically identical plants, right, with all of the same alleles, that depending on the content of the soil that you plant these seeds in, these plants can appear a different color. That is due to the phenomenon known as the norm of reactions. Okay, the norm of reaction is the range of phenotypic possibilities. So the range of physical features that are possible from this single genotype under different conditions. Okay, in this case for the plants, it's the pH of the soil that is the determining factor on what color that these plants are going to be. All right, so the norm of reaction can be quite limited or it can be quite broad, right? So for example, human height, like, yeah, there are genes that influence our height, but once you take into account a number of other factors, the norm of reaction can be very broad. Like, we're all various heights for various reasons. Okay, and so this norm of reaction is quite important to consider. Our genes alone are not the sole determining factor of every phenotypic outcome. Okay, so the impact of the environment on the phenotype cannot be diminished. Okay, the norm of reaction, this example here with the hydrangea flowers of the same genotype, meaning they are genetically the same. They can range from blue to pink to purple, depending on the acidity of the soil. These are the exact same plants genetically. Okay, but due to the environmental conditions, the phenotypic expression of these plants are going to be different. All right, so I'm gonna stop here for today. 2 homework questions that I want you to jot down and follow up with in your notebook. Distinguish between incomplete dominance and codominance. Okay, and then I want you to describe the genetic determination of sex. Okay, how is sex determined from the genetic standpoint? Stop here today.
Just to be clear, we're keeping this in our notebook or we're
That's
submitting it?
correct. That is staying in your notebook.